Hello again. Continuing on the topic of species diversity, we're going to be talking today in greater detail about abundance patterns. As we've emphasized many times in the last few sessions, the abundances of species are not equal, and we've used this or incorporated this into our measure or assessment of diversity. But we can also examine the ways in which these abundances are unequal. There's a classic example of beetles in the River Thames with highly unequal numbers of individuals. The majority of species are represented by only a single individual, while a few species are abundant. And this is actually predominantly the case as a general pattern in communities of different taxa from different parts of the world. Now, we can plot this relationship between species and abundance in many different ways. There are different models in both theory and real data. One classical approach to plotting this is the rank abundance or Whittaker plot. But you can also plot abundance biomass comparisons or ABC curves, frequency distributions or the log series, and the Preston plot or the log normal, each of which we will be examining. The classic rank abundance plot in this instance shows the relationship between four different models, and these are the classic models of relative abundance that were developed more than half a century ago, including the geometric series, the log series, the log normal, and the broken stick. There are actually preferred plots for the different models, including a log of the species sequence, but one may also plot cumulative and relative abundance. Now, before we go into these models, there's an important distinction to make. Many of these are what are called statistical models. They're based on an underlying statistical distribution that approximates the distributions that we see in nature. There's the classic log series and log normal. The negative binomial has also been used to characterize relative abundance patterns, and the Zipf-Mandelbrot model, which has otherwise been used to describe the distribution of words in a text, the relative size of cities, and so on. To emphasize again, these are not based on any underlying biological mechanism. They're based on a statistical distribution. There are also models of species abundance that have been based on underlying processes that are either niche-based or non-niche based. And finally, there's a class of models that we're going to call neutral models that we will also try and understand today. The statistical models, Fisher proposed the log series back in the 1930s. Preston proposed the log normal. And there's, of course, the negative binomial and the Zipf-Mandelbrot that were proposed in the 40s and 50s and developed later on. The log series is closely related to the geometric series. In terms of ecological inference, it's predicted if the intervals of species arrivals in an unsaturated habitat are random rather than regular. The series is given as alpha x squared by 2, alpha x cubed by 3, and so on. And it's derived, the parameters are derived through an iterative solution, and alpha is the parameter that defines the log series. The log normal was visualized by plotting the number of species in different classes of individuals. So if you look at this plot on the what we have on the x-axis for three different for birds in Britain, snakes in Panama, and vegetation in Ireland, is the number of individuals of a particular species. So how many species had just 1 to 2 individuals, how many species had 3 to 4 individuals, how many species had 5 to 8 individuals, and so this is sort of a doubling of each class. You can also have them. You can have a different base, so you can have them as to the power of 10, as you see in the third case, and the log normal distribution produces a shape where the maximum number of species are seen in intermediate classes. So most of the species have an intermediate number of individuals. So again, we have log species abundances on the x-axis. Log 2 involves doubling of individuals, and so on. And it's believed that the log normal may arise as a statistical property of large numbers and a consequence of the central limit theorem, and is given by the following equation, which we won't go into detail, but the things to remember here are that SR, the number of species in the rth octave to the left and right of the symmetrical curve, is given by this equation, where S0 is the number of species in the modal octave. We also have the modal octave of the individual curve that I'll show you in a second. But before we get to that, one of the arguments about the communities following a log normal distribution was that the community overall follows this classical the log normal distribution, but what we see in nature is truncated, and this is because of sampling, and we don't sample the species to the right of what was called the veil line, right? And so if in the plot over here, if we had sampled only this up to this point, we would only capture these species here. As we increase sampling, we capture more species, and moderate amounts of sampling give us these truncated log normal distributions that we see very often in nature, and we very rarely get to a complete sampling that gives us a complete log normal. As I mentioned, one can plot both a species curve, which is the number of species in each individual class, and then one can also plot the number of individuals in the number of actual individuals in those classes. Another example of moth diversity, where we can plot it as where it's plotted as log series and log normal, and one of the arguments is that the shape of the distribution that we infer might even be influenced by the number of amount of sampling that we've done. Therefore, small samples are described equally well by log series and log normal, and that larger samples are better described by the log normal distribution. We touched briefly upon the statistical explanations of the log normal, that is the property of large data sets, a consequence of the central limit theorem. And patterns determined that come about as a consequence of the central limit theorem are often those where there's a very large number of determining interacting factors. Now, there also have been biological explanations. Sugihara in 1980 argued that when a niche space is broken up into species, each time a species invades, a niche is split at random. All niches are equally likely to be selected for division, and that in a system such as this, the community would tend to have a log normal distribution. Other statistical models, such as the negative binomial, have rarely been used to fit species abundance data. The log series itself is a limiting form of the negative binomial, and there's a Mandelbrot that has its roots in linguistics and information theory, has occasionally but not frequently been used in community ecology. But this postulates a rigid sequence of colonists in a community as underlying the pattern produced by this distribution. Another requirement of this is that later colonists have more specific requirements and are rarer. This leads us to another class of models which have an underlying biological process. These are niche-based and non-niche based. The niche-based ones include early models like the geometric series that was proposed in 1932 by Motomura, many that were proposed by MacArthur, such as the particulate and overlapping niche, the broken stick, again proposed by MacArthur, Sugihara's sequential breakage models, and so on. There are also non-niche based models that we'll touch upon very, very briefly. The biological models were summarized very nicely by Tokeshi, and he proposes a series of models that he calls MacArthur fraction, dominance preemption, and so on. And we're going to examine what each of these does. But what's interesting here is that these start with the Motomura's geometric model, which is based on the assumption that ecological communities have niche space which is apportioned between species, right? So the niche basedness of it is that there's a particular space available that gets apportioned. These niches are multidimensional. We're not examining what these niches are composed of. We're not getting into questions of fundamental and realized niches. And we are looking at these assemblages as functional ecological units. And these niche-based models may not be particularly useful when we look at much, much larger assemblages, either spatial or taxonomic. Right? So what's important to distinguish these from statistical models is that we are basing these on certain ecological processes. What are the underlying processes? They can be ecological and evolutionary. They can be niche fragmentation or niche filling. So you can have an existing niche that's broken up or niches that are occupied. These models can be deterministic and stochastic. The geometric series of these is the only deterministic niche apportionment model. Stochastic models often capture random elements inherent in natural processes. And these can result in a range of evenness from very low to very high, and we'll examine the different models that produce that. So the geometric series, in the geometric series, each species preempts proportion k of the resource, and the abundance of each species, n of the ith species, is given by this particular equation, which takes into account the total number of individuals, the parameter k, which is the proportion of the resource, and a constant. The broken stick, proposed originally by MacArthur, has a resource. You can think of it as a stick that's broken randomly into S pieces, produces a more equitable state of affairs, and is usually represented as a rank abundance plot. Again, is given by this form where n is the total number of individuals and S is the total number of species. These are sort of the classic niche models that were proposed. Tokeshi, as we saw before, came up with a number of different models based on the assumption that niche space is sequentially divided among species as they join. The target is divided at random, and most generically speaking, the larger the target niche, the more even the resulting distribution. So how are these models different from each other? In dominance preemption, the smallest niche is always chosen. In random, a niche is chosen at random. There's a power fraction where the niche is chosen at weighted random, and so on. So essentially, the way that the niche is selected is different in each of these models. And each of these produces models of different evenness, and you can go from dominance preemption, which is the least even, to dominance decay, which is the most even. More specifically, to look at these, the dominance preemption, which, to go back a second, is the least even, assumes that each species preempts more than half of the remaining niche. It's conceptually very similar to the geometric series. It can be applied to niche fragmentation, though it's a niche filling model, and produces the least even communities, because, as you can imagine, each species is preempting more than half of the of what's left. So they're taking up a large part of what is remaining in the niche. So the smaller and smaller bits remain. The random fraction is a sequential breakage model that starts with the division of a niche into two pieces, and for further breakage, one of these is selected at random, and this process continues. In this model, new colonists compete for a niche, and this model has been used to cover speciation events as well. Tokeshi's power fraction model is often applied to species-rich assemblages. It's divided initially at random, and the probability of a niche is split is related to its size through a power function. And if k is equal to 1 in this function, this essentially reduces to the MacArthur fraction model, and if k is equal to 0, this reduces to the random fraction model. They actually found that k was equal to more than 0.2 in a range of assemblages, implying that larger niches have a slightly greater chance of being split ecologically or evolutionarily. In the MacArthur fraction model, larger niches are more likely to be divided, which therefore produces a more even distribution of species abundances, and is conceptually very similar to the broken stick, showing that one cannot always use an observed pattern to infer process. Now, this is something also important to remember, that in large ecological studies, we're often trying to derive an ecological or an evolutionary process from the pattern that we see, because it may not be possible for us to conduct experiments at those scales. Cannot put an elephant in a petri dish, and so we're often using the approach of inferring process from a series of patterns that we observe. But some patterns can also be produced by multiple different processes, and therefore one must be cautious when inferring mechanisms or ecological processes from observed patterns. In the dominance decay model, largest niche is always split, and the sizes of resulting fragments are chosen at random. It's a mirror image of the preemption model. If it were deterministic, it would be the inverse of the geometric series and produces a very uniform distribution. There's actually no evidence of this in nature. Tokeshi also proposed a random assortment model where there was no relationship between niche apportionment and species abundances. This might be seen when a community is in a state of flux or undergoing major environmental changes. It's sort of a stochastic analog of the geometric series where k is equal to 0.5, and similar to a neutral model that was proposed by Caswell. The last of Tokeshi's models that's really quite interesting is where he proposes that multiple processes may be involved. Competition is most likely to occur between abundant species, and these divide niche using one or the other of the niche apportionment models. But rare species divide their niche on the basis of random assortment. Now, as you can imagine, this is actually quite an attractive idea, because from everything we know about ecology, it does seem that the species that have larger abundances in a particular area are the ones that are likely to be interacting with each other and competing, and therefore partitioning niches with some underlying process, whereas rare species, the distribution of abundances of rare species may actually be may be random. Of course, this does beg the question of what is rare, and it also begs the question of which of the niche apportionment models should be used. Other kinds of species abundance models have been proposed. Hughes proposed this model where he invoked competition as a structuring mechanism, developed it for marine benthic communities, and in his model has more abundant species than predicted by the log series, but too few rare species. Hughes for the first time incorporated birth and death processes, which as we will see is important when we as more neutral models were developed. Another important step in the development of these neutral models was Caswell's model, who asked what species abundances would be if all biological interactions were removed. Both richness and evenness were lower in the real world than in Caswell's neutral landscapes. And Caswell used this approach to compare observed diversity with expected neutral diversity. For large values of S species richness and abundance, Caswell's model produced values of diversity that resembled the log series. Another reminder again that pattern does not imply process. All of this leads us to Hubbel's neutral theory. Stephen Hubbel extended MacArthur and Wilson's theory of island biogeography to account for regional and local patterns of diversity. MacArthur and Wilson had proposed that the equilibrium number of species on islands would be determined by the processes of colonization and extinction, which were in turn determined by the distance of the island from a mainland and the size of the island or the area of the island. This originally applied to classical islands, but could also be applied to habitats that were insular, such as, for example, the shola patches in the sea of in the surrounding matrix of grasslands that one finds in the upper landscapes of the southern Western Ghats. In Hubbel's theory, he postulated meta-communities as assemblages of trophically similar organisms. This meta-community is composed of sub-individual communities which are always saturated with individuals, and there's a fixed relationship between the number of individuals and area. Now, the extension here of MacArthur and Wilson's theory is that each of these individual communities is isolated from each other. Birth and immigration is equal to death. The relative abundance of species in a community is related to its abundance in the meta-community. He also postulated that species abundance in a community can be shaped by evolution. He introduced the idea of a biodiversity number, which is twice the speciation rate in this community multiplied by meta-community size, and used this to explore consequences of different speciation rates and meta-community size, and found that different models of speciation result in different species abundance distributions. And so Hubbel's theory, or Hubbel's unified theory of biodiversity and biogeography, incorporates the idea of speciation or evolutionary processes as well in producing a species abundance pattern. He showed that point mutations lead to a log series, and that random fission results in random fission of a species into two, results in what he called a zero-sum multinomial distribution, and other outcomes, such as when immigration is unlimited, which is that species can move or individuals can move between these communities in the meta-community. So obviously when immigration is unlimited, the community is similar to the meta-community because there's unlimited movement, and this will follow a log series or a zero-sum multinomial depending on the species mode, right? So when immigration is unlimited, the shape of the distribution depends on the speciation mode, as we just discussed on the previous slide. That point mutations produce a log series and fission produces a zero-sum multinomial. But if immigration is limited, then the community follows a log normal distribution. So the species mode then speciation mode doesn't influence the outcome. That immigration itself limits the nature of the distribution to the log normal. Here, of course, extinctions must be compensated by increases in abundance of existing species, since there are no colonists to contribute rarer species. At intermediate levels of immigration, he found that the distribution is skewed to the left and follows the zero-sum multinomial. Hubbel argued that this accounts for a wide range of empirical species abundance distributions, and this neutrality in this comes from per capita ecological equivalence of all individuals of all species in a trophically defined community. Now, what this means is that in many species abundance distributions, as we saw earlier, we treated all species as equal. The neutrality of Hubbel's theory extends this to the per capita equivalence of all individuals. So none of your individuals belonging to different species are ecologically different from each other in the meta community. And despite this neutrality, the processes of immigration, the size of the meta community, and the mode of speciation itself can produce different species abundance patterns. And to Hubbel, the power of this theory was that assuming ecological equivalence, we could use immigration rates and speciation to produce the patterns that we see in nature and explain the patterns that we see in nature, and therefore infer that these are the primary processes driving these relative abundance patterns that we see in communities. Of course, this has been questioned by many ecologists working on functional diversity. On the one hand, we also know that biological interactions play an important role. So there has been a lot of debate and discussion about this theory. This, of course, takes us back to sort of a recurring theme of biological processes, of which biological processes are important in producing larger patterns. On the one hand, ecologists argue, for example, with the neutral theory itself, that we know that factors such as inter and intraspecific competition, resource use, predation, commensalism, parasitism, all of these processes greatly influence individuals and populations, and therefore, how can we come to arrive at a determination that biological processes are not important, while others argue that while these biological processes might act at certain scales, at much larger spatial scales, spatial temporal taxonomic scales, that in fact these biological processes do not influence the outcomes, and assuming neutrality at that particular, in this case the individual scale, can still produce the patterns that we see, and therefore we don't need to assume those inter-individual differences. The jury is of course still out. So, as we've said repeatedly, it's really important to identify what the goal of the study is. Goodness of fit tests have been used to compare observed and prediction of the model to understand niche apportionment. Diversity has been used to derived from the slope of rank abundance plots. People have compared the abundance of data, the relative abundance of two data sets using, for example, the Kolmogorov-Smirnov two-sample test. So while there are different while there are different models available, while there are different statistical tools available, what we finally use and which approach, which metric, which model is really determined by the goal of the study. Now, very briefly, I'll also touch upon the idea of rarity. We've talked about relative abundance and we've mentioned rare species in the passing many, many times. Now, rarity itself is not just one simple uniform thing. So when we ask the question, what is rare? There are both absolute and relative definitions of rarity. So something can be rare compared to something else, but individual species that have been found only once, there's only a single individual of it has been found, or it's been found in only one of many plots or many samples. These have been defined as rare. In conservation, there's the IUCN's definition of rarity. So ecologists and conservationists often use the term rare to mean quite different things. One approach to look at this is to look at it along three axes. One is geographic distribution, which can be wide or narrow. So widespread species versus narrow or narrow endemic species. Habitat specificity, that can be generalists or specialists, and the population size, that can be either large at least one of the locations where it's found or small everywhere. And as you can see here, there are patterns where geographically widespread species tend to have large populations, at least at one of their locations. If you look at British flora, for example, 36 and 44% of widespread species, while being broad and restricted in habitat, were somewhere large. Now, you get a smaller proportion of species that have small populations everywhere, for British flora at least, and divided across both wide and narrow geographic ranges, and a slightly different distribution of species for freshwater fish, where you in fact find that while you do get broad habitat, widespread species being somewhere large in 29% of the locations, actually the remaining categories, wide and restricted, have both large populations in some areas, but also species, 13% of species that are small in population everywhere. 13% of species that are small everywhere but are narrow endemics, and some that are narrow endemics but have large populations. So this distribution across these three axes can vary both with in different parts of the world and with different taxa, but give us an entirely different way of examining rarity. And so therefore it's important also, even though we've used in our larger discussion of species diversity and relative abundance, we've really talked about it as just rarity, as in abundance, or the number of individuals in a given community. We can think about rarity ecologically along multiple different axes. So in conclusion, different methods of plotting species abundance data were used to visualize these patterns in communities. Both biological and statistical models have been used to understand them. The log series and log normal models were most widely used for several decades to understand relative abundance patterns. Tokeshi's niche apportionment models gives us a really nice framework of all the different ways in which niches can be apportioned, but with an underlying biological process. There have been numerous null models of species abundance, most recently Hubbel's model, and there are different approaches of investigating patterns of species abundance in nature and the statistical and modeling approaches that we use depend very much on the exact questions that we have. And finally, the concept of rarity is more nuanced than just abundance, and it's worthwhile to consider that when we're examining these patterns in ecology. So that's it for today on species relative abundance. But this also brings us to the end of the series of lectures on species diversity and its measurement. Thank you.